Hello and welcome. My name is Kimberly Martin. I am an Applications Chemist 2 and Technical Service Specialist for Waters-VICAM. Today I will be discussing mastering complex samples using Immunoaffinity column sample prep with HPLC and Uplc. In this presentation, we will cover what are mycotoxins, the background of mycotoxins and chemical structures, ecological parameters in mycotoxin production, what ecological factors affect mycotoxins, new solution for the complex matrix, introducing the Aflatoxin WBSR+ column, Uplc and HPLC results and the discussion of those results, and advantages of the Aflatoxin WBSR+ immunoaffinity column. What are mycotoxins? Well, they are the toxic secondary metabolites of several fungal species, including the three shown here: Penicillium, Aspergillus, and Fusarium molds. Aflatoxin is one of, if not the most carcinogenic naturally occurring substance known to man. It has a number of different forms and is found in many common foods like corn, peanuts, tree nuts, and in animal feed components like cottonseed. It can have a huge impact all the way along the food chain. Other mycotoxins, such as Ocratoxin A, occur at much lower levels, but it occurs in a wide variety of commodities, including dried fruits such as raisins, which are an ingredient in a number of common foods such as barley, soy products, which again can be a component of animal feed, and even coffee, and we know how essential coffee is. And that's a concern because although the amounts might be relatively low, it can accumulate in the blood and other tissues and cause other problems. All of the mycotoxins have different impacts, although they are in some way detrimental to either human or animal health, or both. Some are carcinogenic, as mentioned. Some have effects on the immune system. Some might cause types of neuropathy. Others cause things like beak rot in poultry and cause a number of other problems with other livestock. Another mycotoxin, deoxynivalenol, is also known as vomitoxin. That causes problems in many wheat-based products, and we really want to avoid these mycotoxins getting into any part of the food chain. Now we will go over mycotoxin chemical structures. Shown here are the structures for fumonisin B1, B2, B3, and Ocratoxin A. Fumonisins, which were discovered in 1988 in South Africa, differentiate by a hydroxyl group. Fumonisins predominantly occur in cereal grains, especially in maize. Mechanistic studies suggest disruption of lipid metabolism as being the mode of action for fumonisin B1 mediated toxicity. Fumonisin B1 is not mutagenic, but is capable of introducing reactive oxygen species that could lead to DNA damage. Fumonisin B1 caused effects in some developmental and reproductive studies. A slight significant impact of ocratoxin is that it occurs in such a wide variety of commodities, such as raisins, barley, soy products, and coffee, in amounts that may be relatively low. However, significant amounts may accumulate in the blood and other tissues because it derives from potential different sources. Ocratoxin produces necrosis of the proximal tubular epithelium and then is released, whereby it is reabsorbed by albumin and continues to circulate via the bloodstream. Ocratoxin is a carcinogen in rats and mice, and it is suspect agent of a human disease named Balkan endemic nephropathy that affects the kidneys, which tumors are often associated. Other mycotoxin chemical structures are shown here, including T2 toxin, HT2 toxin, deoxynivalenol, nivolumab, and zearalenone. T2 and HT2 toxins are members of the fungal metabolites known as trichothecenes. Corn, wheat, barley, oats, rice, rye, and other crops have been reported to contain the T2 toxin. The major attribute of the T2 toxin and other trichothecenes is that they inhibit protein synthesis, which is followed by a secondary disruption of DNA and RNA synthesis. It can decrease antibody levels, immunoglobulins, and certain other humoral factors. The effects include weight loss or poor weight gain, bloody necrosis or beak lesions, and hemorrhaging. T2 results from an esterification of the hydroxyl group of HT2 with acetic acid. Niv and DON, known as nivolumab and deoxynivolumab, differentiate between each other by the presence of a hydroxyl group in nivolumab. Deoxynivolumab is also known as vomitoxin. Zearalenone is chemically a phenolic resorcilic acid lactone. In fact, it is often present in combination with deoxynivolumab. The compound is mostly found in corn. However, it is found also in other important crops such as wheat, barley, sorghum, and rye. Zearalenone is the primary toxin causing infertility, abortion, or other breeding problems in mammals. Lastly, we have the mycotoxin chemical structures for aflatoxins listed below. As you can see, aflatoxin B1, B2, G1, G2, M1, and M2, which is the primary focus of the immunoaffinity column that I will be discussing today. Aflatoxins are produced by two species of the genus Aspergillus, Aspergillus flavus and Aspergillus parasiticus. Aflatoxins are genotoxic and carcinogenic and can cause both acute and chronic toxicity in humans. Aflatoxin B1 and B2 differ for a double bond, and the same is true for aflatoxin G1 and G2, along with M1 and M2. Corn, cottonseed, peanuts, and tree nuts are main crops affected. The presence innate fluorescent properties. Aflatoxin is the most carcinogenic naturally occurring substance known to man. In summary, mycotoxins are health hazard causing compounds, and it is not a case that they have been ranked as the most important chronic dietary risk factor, higher than synthetic contaminants, plant toxins, food additives, or pesticide residues. One very interesting thing about mycotoxins is that they are not ubiquitous. You may find the mold in your commodity, wherever it may be, and not the toxin, or vice versa. The mold may have been eliminated, but the mycotoxin, while you can't see it, is still there. So why is that? Well, different ecological parameters or stressors affect mycotoxin production by the differential fungal species mentioned earlier, and those include moisture, spore load, the temperature of the environment, if there's any other bugs that have infected the plant, or the storage area, what substrate they are growing on, oxygenation levels, how long something has been stored, maybe some mechanical damage that occurred during harvest, or insect damage. And one important note is that organic crops or crops that have not been treated with fungicides are especially vulnerable to contamination by mycotoxins. So there are many strategies to growers and producers that they can use to minimize risks to the crops and the products. For example, nut growers can reduce the percentage of early split nuts by irrigation management and avoiding tree stress. Early harvest also cuts down on navel orange worm damage. using a rod stock that minimizes early split nuts and carefully use dormancy breaking chemicals can also prevent early splits. Processors can reduce aflatoxin levels by drying nuts as soon as possible after arrival. Hand sorting can remove contaminated in-shell nuts based on the shell discoloration, shell size, and presence of insect frass. Storage at proper moisture levels and avoiding post-harvest rehydration will also prevent contamination. As we have discussed, aflatoxins are carcinogenic mycotoxins that have adverse health effects on both humans and animals consuming contaminated food and feed. A method has been developed for the highly sensitive and selective determination of regulated aflatoxins in a wide range of commodities. The extraction of aflatoxins from representative commodities of interest, corn, wheat, oats, red chili, black pepper, cocoa, and a traditional Chinese medicine, was performed using a solvent extraction, then immunoaffinity column cleanup on the new Aflatest WB SR+ immunoaffinity column. Chromatographic separation was demonstrated using both the Waters HPLC Alliance and the Waters UPLC Acuity H-Class Plus platforms using fluorescence detection, supported with post-column derivatization and large flow cell, respectively. The performance of the method was evaluated through replicate analysis of spiked test portions of 7 different matrices. Overall, recovery was shown to be excellent between 92 and 116%, with relative standard deviations lower than 8%. The method was found to be specific as no interference peaks were observed for blank samples. The method has been demonstrated as suitable for monitoring compliance with regulatory limits set for aflatoxins in food commodities globally. We have discussed mycotoxins in detail and why we need to test for aflatoxins in our food supply. A variety of testing solutions exist for the determination of aflatoxins, ranging from easy-to-use rapid test, which can be used at the point of production, to lab-based reference methods that are more time-consuming but can be used to provide a more comprehensive view of the level of contamination. Determination of aflatoxins is a challenging task due to the complexity of the matrix, often with high lipid content, the low concentrations in which these compounds are usually present, and the strict low regulatory limits. The combination of cleaning up using immunoaffinity columns based on antibodies and analysis by HPLC with fluorescence has been a prerequisite step to achieve the desired sensitivity and selectivity, and has been successfully used as a cost-effective way to check compliance with the regulatory limits for aflatoxins for many years. VICAM launched a new product, the Aflatest WB SR+ immunoaffinity column, which can endure a high concentration of organic solvent while maintaining excellent recoveries. The objective of this study was to examine the performance of using VICAM's Aflatest WB-SR+ immunoaffinity columns with 2 separate HPLC-UPLC platforms in both grains, corn, wheat, and oats, and difficult food matrices such as spices, chili powder and black pepper, along with cocoa and a traditional Chinese medicine, using a standardized testing procedure. In addition to the Aflatest WB-SR+ immunoaffinity column, it was evaluated for its ability to bind sterigmatocystin, which is another potent liver carcinogen and a precursor to aflatoxin. In the results shown here, different concentrations of acetonitrile were passed over the column at different spiking levels. This chart shows acetonitrile effect on Aflatest WB-SR+ recoveries. And as you can see in this, for the 2 nanogram mix and the 500 nanogram mix, there are very excellent recoveries along with great % CVs. The Aflatest WBSR Plus column was also compared with our competitors' columns using increased solvent tolerance. We saw that Aflatest WBSR Plus can tolerate up to 40% methanol and 30% acetonitrile over the columns. This is based on 100 nanogram total aflatoxin spiked into 20% acetonitrile and a PBS solution. As shown here, you can see that G2 recovery was very good for the Aflatest WBSR Plus column, and the competitors had low G2 recoveries, along with one competitor that had a low aflatoxin B2 recovery. Shown here is the linearity and LOD and LOQ for spiked corn samples. As you can see, the LOD and LOQ are within regulatory limits for both the United States and the EU, and the linearity showed recoveries for the 2 nanogram to 500 nanogram from 96% to 100%, along with linearity of an R squared of 0.9999, which is excellent. As mentioned previously, the Aflatest WBSR Plus immunoaffinity column is also capable of capturing the precursor to aflatoxin, STC. The STC recoveries in spiked corn samples are shown here. The structure is also shown next to the peak in the UPLC chromatogram from the analysis of an extract of corn spiked with 25 micrograms per kilogram of STC. The recoveries ranged in levels spiked from 4 to 50 micrograms per kilogram, from 92% to 98%, and the CVs also were below 8%, which is very good for this analysis. Shown here is naturally contaminated corn using a 90% acetonitrile extraction and an 80% methanol extraction. And in comparison, you can see for the higher level naturally contaminated corn, the trilogy result that we had received was 301 parts per billion, and when we ran this using our extraction, we received a 301.3 ppb result. With the methanol extraction, you can see that it was a bit lower for the result compared to trilogy's result, which was 280 in comparison to the 301 ppb. This shows that higher levels of organic extraction clearly extract aflatoxin out better, and also with our column being able to tolerate the higher solvent, it makes for an overall better extraction with immunoaffinity cleanup. So one method was used for a complex sample matrices analysis. 10 grams of sample was weighed out, and 100 mils of 90% acetonitrile water was added to that into a blender and blended for 3 minutes. It was then filtered. 10 mils was diluted with 30 mils of 2% tween PBS. 10 mils is passed over the Aflatest WBSR Plus immunoaffinity column. The column was then washed with 2 times of 10 mils of water, and it was eluted with acetonitrile methanol at a 1:2 ratio. The extract was then diluted and injected onto the HPLC or UPLC. Shown here are the results for wheat, oats, cocoa, black pepper, and chili powder in respect to their spiking levels. AOC recommended recoveries were 40 to 120 for the lower levels and 60 to 115 for the higher levels, in which we fell into the recommended range for all of our matrices using this method coupled with the new immunoaffinity column. Traditional Chinese medicine was also extracted using this method and the new column, the Aflatest WBSR Plus immunoaffinity column. And the Fallopian multiflora results are shown below. Spiked at 5 micrograms per kilogram and 20 micrograms per kilogram, the AOAC's recommended recoveries are 75 to 120 for the 5 microgram and 70 to 125 for the 20 microgram per kilogram. the RSDs were all in range and the recoveries were all in range, ranging from 93 to 105%, with the highest RSD being a 4.1, which is well within the limits for the AOAC recommendations, and also showed that this will work for traditional Chinese medicine as well as spices and oats and corn. Shown here are the HPLC conditions for aflatoxins. We used an Alliance E2695 with a fluorescence detector that had a photochemical reactor. We used a Novapac C18 Waters column. The column temp was 25 degrees C, sample temp was 25 degrees C. The injection volume was 50 microliters. The flow was 0.8 mils a minute. And the mobile phase was water methanol 5545 isocratic. The software was Waters and Power software for the system. The UPLC conditions for aflatoxins are detailed here. The LC system was a Waters Acuity UPLC H-class with FDN sample manager. The detection was an Acuity fluorescence detector with a large volume flow cell. Excitation was 360, emission was 440. The column that was used was a UPLC Acuity HSS T3 column. Column temp was 25 degrees. Sample temperature was 25 degrees C. Injection volume was 3 to 6 microliters. And the flow rate was 0.3 mils a minute. The mobile phases were 5545 water methanol isocratic. And the software that was used for data reduction was Waters and Power software. HPLC and UPLC chromatograms are shown here. The one on the left, HPLC chromatogram with the red and the green lines. The red line is unspiked black pepper and the green line is spiked black pepper with 4 ppb total aflatoxins. And as you can see, there is no matrix interference in the blank sample. The UPLC chromatogram on the right is from the analysis of an extract of Fallopian multiflora spiked with 5 microgram per kilogram of total aflatoxin. And shown below that is the Aflatest WBSR Plus column. Some of the advantages of the Aflatest WBSR Plus Immunoaffinity column are listed here. High performance. The Aflatest WBSR Plus method exceeds AOAC method performance requirements, giving greater than 90% mean recovery of aflatoxins B1, B2, G1, and G2 in both grains, corn, wheat, and oats, and difficult to analyze food matrices such as chili, cocoa, and traditional Chinese medicine. The column and the method is comprehensive. VICAM's Aflatest WBSR Plus Immunoaffinity column binds aflatoxin B1, B2, G1, and G2, as well as aflatoxin M1, M2, and STC. The method is flexible. It's one standardized method that is suitable for a wide range of different commodities. It also has a wide testing range. Aflatest WBSR Plus can detect total aflatoxin levels from 0.05 nanogram to 500 nanograms, with recoveries greater than 90%. The Aflatest WBSR Plus method exceeds AOAC method performance requirements and shows that it is capable of getting excellent recoveries on both grains and complex sample matrices. It is fast and easy. The UPLC option provides an opportunity to shorten the analytical runtime, and the use of a large flow cell in the fluorescence detector eliminates the need for post-column derivatization. And lastly, we have overall performance. These methods have the desired sensitivity, selectivity, and overall performance that is used to check compliance with regulatory limits for aflatoxins in a wide range of crops and foods. What VICAM delivers. VICAM delivers testing options and programs to monitor mycotoxins in a wide variety of commodities. We are dedicated to ensure that our food supply is mycotoxin-free and safe for human and animal consumption. I would like to thank you so very much for joining me today and for your interest in the new technologies for complex matrices and solving some of the difficult challenges that occur when analyzing difficult matrices. I hope that you have found this particularly helpful and that you will join us in the future for other topics. Thank you so much.